We have heard of some microbes that have the ability to cause pandemics. In the recent times, we have witnessed the COVID 19 pandemic. But however, if I have to tell you of one microbe that has successfully caused about 7 pandemics across the globe, it is Vibrio Cholerae. So, in today's session, as we learn about Vibrio Cholerae in detail, let me begin by taking you through a case. This is of a 10-year-old boy who was brought to the casualty with a history of passing watery stools for the past 2 days. So, history of 2 days of loose stools. His parents informed that the child had vomited once and also has muscle cramps. However, the child has no fever and no abdominal pain. On examination, the child is afebrile, the eyes were sunken. The pulse, please note, was 112 per minute, suggesting of tachycardia. Skin was cold and clammy, and blood pressure was 80 by 60 millimeters of mercury. Again, probably hypotension. Abdomen was non-tender. So, if I have to summarize, this is the case of acute diarrhea. So, as we progress through studying this pathogen, you can attribute Vibrio Cholerae to be one of the important differential diagnostics for a case of acute diarrhea. So, if I have to introduce Vibrio Cholerae to you, you must know about the epidemiology and natural habitat of this pathogen, cultural and biochemical properties, pathogenesis of infection, clinical manifestations, the diagnostics, and prevention and treatment. So, let's begin by understanding that initially there was a belief in the earlier days that cholera was spread from one person to another, or probably a person got it himself by breathing vapors or bad air because of some evil in the air or miasma in the atmosphere. It was only when Dr. John Snow put forward the theory that cholera is a waterborne infection after he thoroughly investigated the Broadway Street pump outbreak in London, and this outbreak involved almost 600 cases which were positively tested for cholera. And it was Robert Koch who isolated this organism for the first time in 1886, and it was named at that time as Coma bacillus because it is a coma-shaped bacterium, and based on that it got its name. So now let me describe the morphology of the bacterium. Vibrios are highly motile. They are gram-negative coma-shaped rods with a single polar flagellum, and in a stained mucus flakes from stool samples, these Vibrio Cholerae appear to be arranged in parallel rows, resembling a school of fish appearance. So if you take this as a microscope slide and you do a gram stain, probably they will have a school of fish appearance, appear to be arranged in parallel lines to each other. And where do they survive in nature? What is their epidemiology like? So Vibrios are ubiquitous and found worldwide. The Vibrio Cholerae specifically can survive in water, in marine environments, both seawater, in seafood, surface waters, river, in sewage water, and also sometimes in association with aquatic animals. There are causes, these are the causes for endemic and epidemic outbreaks in areas where you find poor sanitation, because the world has witnessed 7 global pandemics of cholera infection in the past, and it's very common in developing countries. So this is the background epidemiology, which also highlights to you the public health importance of cholera as an infection. So coming to the classification of Vibrios, Vibrios can broadly be classified into 2 groups, that is the halophilic or the salt-loving group, the non-halophilic or those which do not totally depend upon high concentrations of salt. So among the halophilic or the salt-dependent Vibrios, these are the ones which require high salt concentration of 7 to 10% for their survival. And important examples in this group are Vibrio Parahemolyticus, Vibrio Alginolyticus, and Vibrio Vulnificus. Whereas in the non-halophilic group, we have those Vibrio which grow without extra added salt. However, please note, they'll be happy if you can give them 1% of sodium chloride. And here we have the Vibrio Cholerae and Vibrio Mimicus. So there have been several attempts made in the past to classify Vibrios. One such attempt was by Herberg, and this classification was entirely based on the fermentation of mannose, sucrose and arabinose, that is sugar fermentation, based upon which the Vibrios were classified into 8 groups, and the Vibrio Cholerae was a part of group 1. Whereas in more recent times, as the antigenic structure of the bacterium became known, the Vibrios were classified by Gardner and Venkatraman. And this classification is based on the somatic O antigen and the flagellar H antigen of the bacterial cell. So let's see this classification in a detail. This is the Gardner and Venkatraman classification that is currently followed worldwide for classification of Vibrios. So here under this you have the entire genus Vibrio, and then you have the Vibrio Cholerae and the other Vibrio species. The Vibrio Cholerae is again classified into various subgroups, or we also call them as serogroups or serovars. So we have several serovars up to 200, of which serovar O1 and O139 are very, very important because of their disease causing ability. Whereas we have the non-O1 and non-O139 which do not cause life-threatening infections. So this is based upon the somatic O antigen. Next we have the biotypes. Among these, that is among the O1 subgroup, we have 2 biotypes, that is a classical and the LTOR biotype. And these biotypes, as it is indicated, are classified based on the differences in their biological characteristics. So classical biotype and the LTOR biotype. Now among both, we have 3 serotypes each. So among the classical also there are 3. Among the LTOR also you can get 3 serotypes. That is Ogawa, Inaba and Hikojima. So identification of the serotypes is based entirely on serotyping. Next, let's discuss in detail about the O1 group, because I told you it is the important serogroup that is associated with infections. So O1 group is agglutinated by O1 antisera, that's why the name, and it's responsible for all the pandemics and most of the epidemics that the world has witnessed. And only O1 and O139 are toxigenic. Again, cholera is a disease that is mediated by the production of a toxin. So there are 2 important serogroups, that is O1 and O139, which have the ability to be toxigenic. So let's look at serogroup O139. Since 1992, it has caused several epidemics and outbreaks all along the east coast of India and especially in Bangladesh. Then you have the non-O1 and non-O139 serogroups, which cause occasional sporadic outbreaks. However, none of them have been associated with the epidemics of cholera. So coming to O139, this was first isolated from Chennai in the year 1992. And then on O139, non-agglutinated by any of the antisera, which was between O1 and O138. So we got the new serogroup called as O139. It was for a long time very famous as the Bengal strain because it was from here that it rapidly spread across the coastal belt of East India along the Bay of Bengal. It's also known to cause invasive bacteremia and extraintestinal manifestations, whereas there is no cross protection between the O1 and O139 strains. But you must remember that by 1994, that is within 2 years after its first isolation, the O1 LTOR replaced O139. So this is the highlights of O139, and then we need to know what are the differences between the biotypes. As I told you, there are 2 biotypes. One is the classical and another is the LTOR biotype found in O1. So let's see what is the biological differences between these 2 biotypes. Most importantly, it is the presence of beta hemolysis on sheep blood agar, which is positive in the LTOR biotype, not seen in the classical biotype. Then we have the agglutination of chick RBCs, again a highlight of LTOR biotype. And if you take The susceptibility to an antibiotic called polymyxin B, 50 international units, the classical biotype is susceptible. Similarly, the classical biotype is also susceptible to group 4 phages. So when you use the group 4 phages, they can act on this classical biotype but not on the LTOR biotype. Then we have the Vox-Prosker test. It is a biochemical reaction that is positive in the LTOR Vibrios. And finally, the cholera toxin producing gene. It is a Ctx1 gene that has been identified in the classical biotype and the Ctx2 gene that is found in the LTOR biotype. So now let's look at the epidemiology of cholera and its importance as a disease of public health. So cholera is, you must remember, a notifiable disease. Any case occurring in the community has to be notified to the government, and it's quite often underreported. So the rough estimate of cases that we have is that across the world we have about 1.3 to about 4 million cases of cholera each year, contributing to about 21,000 to 1.4 lakh cases of death. And majority of the cases worldwide are due to O1 LTOR. So West Bengal, which was initially for a long time considered to be a home of cholera, now does not hold its forte because you can see all the states that are affected after that. And both morbidity and mortality have greatly reduced with time. And we have a very good center, that is a National Institute of Cholera and Enteric Diseases, which is a referral center for the Vibrio Cholera. So a lot of work is being done here. You must know that cholera between the epidemics keeps persisting. Between the epidemics, it is maintained by carriers and subclinical cases. And when there is no epidemic, that is in between two epidemics, it is maintained in environment in the seawater. So that's how it keeps itself viable and ready for the next outbreak. Looking at the cultural characteristics of this very interesting microorganism, you must know that it can grow in a wide range of temperature and the pH range for its growth. The best pH range, however, is 7 to 9. That is, it prefers an alkaline pH. It also serves as enrichment condition for the bacterium, and temperature range is quite wide, 18 to 37 degrees Celsius. The Vibrio Cholerae especially, you must know, can grow without salt because they are not halophilic, but you have a separate set of halophilic Vibrios as well. This is how the Vibrio Cholerae looks like when you cultivate it in the laboratory. If you're trying to grow it on nutrient agar, it gives rise to transparent moist colonies with a bluish tinge, as you can see the individual colonies here. And on Mekongki agar, it will appear pale pink non-lactose fermenting colonies. You must remember Vibrio Cholerae does not ferment lactose. So you get non-lactose fermenting pale colonies on Mekongki agar. Similarly, let us look at the characteristic features of the bacterium when it grows on blood agar. It produces a characteristic greening around the colonies. We do not typically call it as hemolysis because it's a full zone which is seen all around the growth, and this is known as hemodigestion. So this phenomena is known as hemodigestion, a hallmark of Vibrio Cholerae seen on blood agar. Also, there are some transport media which are very important for the transport of samples when you are suspecting a case of cholera. And some of the important transport media are Venkatraman Ramakrishna medium, it's also known as VR medium, Carey Blair medium, or even if you do not have any of these, you can go for autoclaved seawater. Then of course there is enrichment medium. This enrichment medium is very important since we are using the stool sample as a specimen. And as you know, stool will have normal bacteria, normal commensals of the intestinal tract like E. coli, and this enrichment media exactly tries to suppress it and enhances the growth of Vibrio Cholerae. And one of the important media which acts as enrichment is alkaline peptone water. You must know that Vibrios survive and replicate in high pH. That's what they prefer. And other organisms are killed whenever you increase the alkalinity of a pH of a medium. So this is a selective media which is used for growth of Vibrio Cholerae, and we call it the thiosulfate citrate bile salt sucrose agar, which is very famously called as the TCBS agar. And here it has a sugar called sucrose, and based on the fermentation of sucrose, you will get yellowish discoloration of the media if it is growing Vibrio Cholerae. Whereas if any other bacteria which is not fermenting sucrose is growing, you will get black colored colonies on TCBS. Right. There are a few biochemical reactions which also help in the identification of this culture that has grown on the agar plates. Vibrio Cholerae specifically is catalase and oxidase positive. Both the tests positive, and it ferments glucose, maltose, mannose and sucrose, but most importantly does not ferment arabinose and lactose. That's very important to note. And this fermentation is for production of acid only. There is no gas production by Vibrio Cholerae. It is also indole test positive. That means it produces indole from peptone. Nitrates are reduced to nitrites. And there is an important reaction which is known as Cholera Red reaction. You can elicit this reaction by adding a few drops of concentrated sulfuric acid to an overnight or a 24-hour peptone water culture of Vibrio Cholerae, and upon adding this sulfuric acid, you will get a red color. So this is a positive Cholera Red reaction. Let us go to understand cholera as a disease. Cholera is a life-threatening secretory diarrhea because of the toxin produced by Vibrio Cholerae. And as I've already mentioned, it has a great potential for causing epidemic and endemic outbreaks. So how does this organism enter into the human body? It is mainly through contaminated food and water. And in the context of having inadequate water sanitation and sewage treatment. So it's commonly a disease of the developing countries and not more often found in the developed world. Improperly cooked shellfish can also sometimes harbor Vibrio Cholerae. So this type of a disease may be found even in the developed countries. So you must remember both Vibrio Cholerae O1 and O139 are capable of producing the cholera toxin, and cholera toxin resulting in clinically acute case of diarrhea. So these are the 7 pandemics as you see from early in 1817 up to the 21st century, which have been caused by Vibrio Cholerae. This disease has traveled almost across the globe. So a great potential to cause pandemics. So the 7th pandemic is really noteworthy because it started in 1961. And it's quite different from the 6 pandemics the world saw earlier, because it was the only pandemic that originated outside India, and all other pandemics prior to that started in India. Whereas the 7th pandemic started in Indonesia in 1961, and India was affected 3 years later in 1964, and it circumscribed the whole world by 1991. Of course, it lasted more than 3-4 decades, and it's the only pandemic to be caused by L-tor. So the biotype L-tor was involved in this pandemic. Right, so moving on to the clinical features of cholera disease, the incubation period after consumption of contaminated water is about 2 to 3 days. So very short incubation period, and a high infectious dose is required. That means at least 10 to the power of 8 bacteria are required to overcome the gastric barrier posed by the acidity in the stomach. It begins with abrupt onset of vomiting and life-threatening watery diarrhea. When I say life-threatening watery diarrhea, it is about 15 to 20 liters of diarrhea per day. And as more and more fluid is lost, feces becomes streaked with stool, and later the consistency completely changes to what is called as rice water stool, which is the hallmark of cholera. So this rice water stool is usually colorless, odorless, and it has no protein contents, hardly speckled with some mucus. So this is the important feature of the diarrhea caused by cholera. And you must also know that crossing the protective layer of mucus, how this organism actually progresses to cause disease. So this describes the pathogenesis of cholera. It is, as I told you, having a single polar flagella. So because of its single polar flagella, It is a highly actively motile organism. It uses this motility to its advantage. It also secretes mucinase and other proteolytic enzymes to overcome the mucin secretion. And it also secretes hemagglutinin protease. It's also called as cholera lectin, which cleaves the mucus and the fibrinonectin. For adhesion and colonization of this organism is facilitated by a special type 4 fimbriae called as toxin co-regulated pilus. This is very important. As you know, pilus is nothing but fimbriae. These are organelles which are required for adhesion to the host cell. And finally, the most important hallmark of cholera is the cholera toxin. Cholera toxin is again an exotoxin. It is a heat labile exotoxin of the AB type. So these are some of the important virulence factors that this organism harbors to overcome the host defenses and establish itself as a pathogen. So let's see the mechanism of cholera toxin. As I told you, it is a dual subunit toxin. You have the A subunit, which is the active one, and the B subunit, which is required for the adherence of the toxin to the intestinal mucosa. So once you have the adherence caused by the B subunit, there is internalization of the A subunit. So here you have the A subunit, which is now internalized, and this A subunit goes and causes ribosylation. So ADP ribosylation of the G protein. The moment the G protein gets ribosylized, immediately it will lead to activation of adenylate cyclase, because of which there is tremendous increase in cyclic AMP, almost 1,000 times increase in cyclic AMP, and because of which there is opening up of the ion channels. So when I say ion channels are opened out, the person will lose chloride, the person will lose sodium from the intestinal wall along with water. So this is the basis for the diarrhea that you see in a case of cholera. So the fragment A is the active fragment and fragment B is required for binding, and the main hallmark of pathogenesis is the G protein ribosylation, leading to increase in cyclic AMP, activation of ion channels, and ions getting flown out along with water. So this leads to diarrhea and dehydration. So let's see a little more about the cholera toxin, which causes induction of cyclic AMP, and loss of fluid and electrolytes can lead to shock due to profound dehydration. It can also lead to acidosis. Now you may ask me, where does the gene to code this cholera toxin come from? The gene which codes for cholera toxin is called as the Ctx gene, and this cholera toxin is a phage mediated toxin. So that means it comes from a bacteriophage. You know of even the diphtheria toxin, which is again a phage mediated toxin. It is also a AB type of a toxin. So this phage genome, as it encodes for the Ctx gene, it also encodes for the TCP gene. As I told you, TCP is important for production of a pilus, and this pilus helps in the adherence of the bacteria to the mucosal cells. So let's go look at the clinical manifestations of cholera. Approximately 75% of the cases may remain asymptomatic, whereas 20% of them may have mild diarrhea. It is those 5 to 10% of the cases which have an explosive, life-threatening diarrhea, characterized by rice water stools, and vomiting may be present, but fever is usually absent in a case of cholera. So complications in cholera are mainly attributed to two things. One is loss of fluids, another is the hypokalemia due to loss of ions. So hypokalemia due to low levels of potassium in the blood can lead to cardiac arrhythmias and renal failure. Similarly, the cholera toxin blocks the uptake of sodium and chloride, thereby leading to dehydration, hypovolemic shock, metabolic acidosis. These are some of the complications in a case which is not treated appropriately in time. So let's move on to understanding the laboratory diagnosis of this infection. The sample that has to be collected obviously will be the stool sample. So if a patient has got rice water stools, that has to be collected. If the patient is suspected to be a case but not actively having diarrhea, You can go for collection of rectal swabs, or a blotting paper soaked in stool sample is also fine to be a specimen of choice. Next is transport medium. As I told you, you require a transport medium like Venkatraman Ramakrishna medium or Carey Blair medium to transport the sample to the laboratory. Once the sample reaches the laboratory, first thing that will be done to look for cholera is a hanging drop preparation. That is, we take a slide and we take a cover slip on which we place one drop of the stool sample, that is rice water stool, invert the cover slip on the slide, and then see it under the microscope. What we look for is the characteristic darting motility shown by Vibrio Cholerae. They just dart across the field. It is like a shooting star type of an appearance. So we can also visualize these bacteria by doing a dark field or a phase contrast microscopy, where because of the darting motility, bacteria will appear like shooting stars. However, if you do a gram stain from this stool sample, you will see gram-negative comma-shaped short bacteria. Typically, fish in stream appearance may be present. Now let's go to the culture aspect of cholera. As I told you, for culture, you will have to first use an appropriate transport media, get the sample to the laboratory, and then perhaps use an enrichment broth to bring out the growth of cholera, suppress the growth of other commensals. So alkaline peptone water is a good enrichment medium. Then you can choose a selective medium so that from this you can subculture the media onto a selective agar. And one of the important selective media is TCBS and Mansour's GTT agar. Mansour's GTT agar consists of gelatin, taurocolate and tellurite in that. And TCBS agar you have already seen. It is taurocolate citrate bile sucrose agar. So the stool sample is plated on semi-selective and selective media. Media can also be plated after enrichment of about 6 to 8 hours and then put on a selective media. Now once you have got the growth on media, we are supposed to identify the culture. So to identify the culture, we will do it on the basis of colony characteristics on the media and some of the important biochemical reactions of this particular bacterium. And finally, we can identify by an agglutination test with appropriate antisera, whether it's the O1 serogroup or O139, or whether it is the serotype in Ogawa, Inaba, Hikojima in that way. So looking at the cultural characteristics, these are the non-lactose fermenting colonies that we get on a McConkey agar. On blood agar, the hemodigestion, this is the hallmark of Vibrio Cholerae. And of course, on TCBS, the sucrose fermenting yellow colored colonies, which are suggestive of Vibrio Cholerae. So there's another way of identifying from these cultures. A positive string test is used to separate Vibrio species from Aeromonas species, because Aeromonas is another bacterium which is also oxidase positive. That's why to separate out the Vibrio and identify it from Aeromonas, we use the string test. This test is done by taking a 18 to 24-hour Vibrio culture, which is then emulsified with sodium deoxycholate or sodium taurocolate. Both of these are bile salts. And it lyses the cell wall of the bacterium, releasing the DNA. So as you pick up from this solution, as you pick the colony up with the help of a loop, you will see the formation of a string. So this is referred to as a positive string test, and this is seen in Vibrio. So this is an indirect indicator that your culture may be belonging to the genus Vibrio. Other than this, there are a few other tests that will help you to identify. Biotyping can be done on the culture that you have grown to differentiate classical and Eltor types. You have seen what are the differences between the biotypes. Serotyping can be done to differentiate the serogroups, whether it's O1 or O139, which we are interested in. And also serotyping can be done to differentiate the 3 serotypes, that is the Ogawa, Inaba and the Hikojima of the serogroup O1. Antigen detection can also be done using a cholera dipstick assay. Though we don't readily do this, we still rely on culture and biochemical tests. And molecular detection can be done directly from the stool sample if there is a facility for multiplex PCR to detect common pathogens causing diarrhea. Also, my antimicrobial susceptibility testing can be done. And with this, we end the lab diagnosis of Vibrio Cholerae. Now, going to the treatment aspect, even before identifying what is the cause for this acute diarrhea, here there is one catch. It is very, very important that rehydration therapy should begin immediately. Don't wait for the report coming from the laboratory. The first concern here is to rehydrate the patient. You can use oral rehydration solutions. You can use IV rehydration therapy. And you may ask, what is the role of antimicrobial therapy? We all know that this is a toxin-mediated infection. Yes, antibiotics, if given, they can hasten the clearance of the organism from stool so that there is no more toxin production. And drug of choice, in case you are giving an antibiotic, is macrolides. We can give azithromycin, erythromycin, or even tetracycline or doxycycline to hasten the process of clearing the bacteria from the gut. So next, coming to preventive strategies, general prevention measures like safe water for drinking and sanitary disposal of feces, the sewage treatment, proper food sanitation go a long way in protecting against outbreaks. And also, in case there is a primary case, prompt outbreak investigation can actually retard the transmission of the disease. It's a very important to notify about the disease, and health education to the general public plays a very important role. Chemoprophylaxis also has a role to play. Tetracycline is usually given to all the household contacts wherever the case is identified, but this should be followed only during epidemics, as not as a norm otherwise. Next, there are cholera vaccines, and there are basically two types of vaccines, injectable vaccine and the oral vaccine. First, let's discuss about the injectable vaccines. These were used in the past, but because of the adverse effects that are associated and because there is no local immunity that they induce, they are no longer used. They have discontinued the usage of injectable killed vaccines for cholera. Now what we have is the oral cholera vaccines. When I say oral cholera vaccines, we have two types. One is the killed whole cell vaccine, and here you have the whole cell vaccine and whole cell recombinant B subunit vaccine. So there are two variations here, and in the live we have the live oral vaccine that is currently available for Vibrio Cholerae. So let's look at the whole cell vaccine first. So when I say whole cell vaccine, it is composed of the whole killed cells of the cholera bacterium. So Vibrio Cholerae, the entire cell is used in vaccine production of the serogroup O1 and O139. It is given as 2 doses, given orally, and with minimum of 2 weeks gap between the dose. For all individuals above the age of 1 year, you can give this vaccine. And the protection it offers is for about 3 years. We have a more advanced format of that, and this is known as the whole cell recombinant B subunit vaccine, where a subunit causing the cholera toxin, B subunit, is also included in the vaccine. So the schedule of this is 2 doses are given orally and with minimum of 1 week gap between the 2 doses. A third dose is given especially in children aged between 2 to 5 years, and protection offered by this vaccine is roughly for 2 years. Next, we have the oral live attenuated vaccines. Live attenuated cholera vaccines given orally using the strain CVD103HGR. Now, if you ask me, this belongs to O1 type. This is a classical biotype, and it is Inaba serotype. So this is the vaccine strain that is used, and the indication is it is recommended for adults in the age group of 18 to 64 years and traveling to those areas where you have active cholera transmission going on. The protection it gives is about 90% at about 10 days after vaccination, and it's a short-term protection, lasts for about 3 to 6 months. So now let's discuss in briefly about the non-O1 and non-O139 types of Vibrio Cholerae. So you know that biochemically they resemble Vibrio Cholerae, but they do not agglutinate with the O1 nor the O139 antisera. They usually do not cause Vibrio Cholerae pandemics or epidemics, but commonly they are associated with gastroenteritis, especially after seafood consumption, that is mainly raw oysters, where this bacterium could be harbored. So you'll get watery or very poorly formed stool, There could be bloody or mucoid diarrhea as well. Abdominal cramps, nausea, and vomiting along with fever could be present. Extraintestinal infections sometimes do occur, especially when people go for recreational activities to seawater. So, otitis media, wound infections, and very rarely bacteremia. So, this is because of either occupational or recreational exposure to seawater, which can harbor these type of Vibrios. These are all sensitive to tetracycline, ciprofloxacin, third generation cephalosporins, which can be used to treat these infections. We will now look at halophilic Vibrios. I told you of this group of halophilic Vibrios which love to survive in high salt concentrations. So they would love to survive in salt concentrations as high as 6%. You can find them widespread in marine environments, and they tend to occur mainly during late summer and early rainfall when the bacterial counts of these bacteria are highest in water. So let's first discuss about Vibrio parahemolyticus. It is one of the halophilic Vibrios. Vibrio parahemolyticus causes foodborne gastroenteritis following the consumption of uncooked seafood, again oysters. Watery diarrhea or dysentery can occur. Rarely one may get extraintestinal manifestations such as otitis media, wound infections. In the laboratory, this microorganism can be identified by this phenomena called as Kanagawa phenomena. An interesting one where beta hemolysis is seen on Vagatsuma agar. And Vagatsuma agar is a special type of high salt blood agar. So basic blood agar where you have a very high salt content. That is to enable the halophilic Vibrios to grow and suppress the growth of other organisms. So swarming sometimes can occur on blood agar caused by Vibrio parahemolyticus. Another important halophilic Vibrio sometimes associated with human infections is Vibrio Vulnificus. We do have some cases of primary sepsis that have been reported, especially in patients with underlying liver disease or those who are having iron overload, or rarely in those having renal insufficiency or who are on immunosuppressives. So primary wound infections caused by Vibrio vulnificus are also present. So it can cause painful erythematous swellings or cellulitis or even vesicular or bullous necrotic lesions, and it affects people usually without any disease. So commonly sepsis is found in those who are immunocompromised, whereas the primary wound infection can be seen in anybody. And vulnificus is a Latin word for wound maker. So it has a predilection to cause primary wound infection. We also have Vibrio alginolyticus, which is another halophilic Vibrio. Occasionally, it can cause eye, ear, and wound infections, again acquired from seawater. So now let us summarize what we learned in this session. We learned about Vibrios and their marine habitat. We learned about the epidemiology and the disease-causing pattern of Vibrio Cholerae, where we know that it can cause epidemic outbreaks, pandemic, and even endemic cases of cholera. We learned about the cultural and biochemical properties. We saw the growth of the organism on blood agar, on nutrient agar, on TCBS, which is a selective media. Another important selective media is GTT, so gelatin taurocolate tellurite agar. Pathogenesis of cholera is mainly mediated by the cholera toxin, which is a AB subunit toxin. A is the active component which causes increase in cyclic AMP secretion inside the intestinal mucosa, thereby leading to severe diarrhea characterized by rice water stools, leading to dehydration. So diagnosis involves microscopy, especially the hanging drop preparation, and gram stain, where you can see comma-shaped bacilli. Darting motility is the highlight on hanging drop. And finally, if you can grow it in selective media, this culture can also be identified by serotyping. Coming to the treatment of the condition and the preventive aspects, rehydration is the mainstay of treatment. Antibiotics can be given to clear the bacteria from the gut, and we have cholera vaccines that are available. We have the whole cell vaccine, and we also have the oral live vaccine. So that's all we have in this session, where we have extensively discussed about Vibrios, their classification, the disease caused, as well as prevention strategies.